Hello everyone, I am Siddharth and welcome to my video. Today we will talk about genome editing. The areas we are going to cover today are what is genome editing, history of genome editing, and applications. So without further delays, let us get into the video. Genome editing is a method used by scientists to edit the genes of an organism to bring about changes in its physical characters. They use scissors to cut at specific spots of a DNA to add, remove, or replace a gene. Scissors here doesn't mean the literal scissors we use normally. They are commonly called restriction enzymes. The history of genome editing dates way back to the late 1900s. Two techniques were used before the current CRISPR-Cas9 scissors. The ZFN method, in its elaborate form known as zinc finger nuclease, are fusion proteins that recognize and bind specifically to 3 to 4 base pair long target sequences. They use the protein DNA binding sites. The next one is the TALEN. TALEN fusion proteins are designed to bind to specific target sequences that flank target sites. Now, if you don't know what flanking means, it just means that the DNA sequence that is adjacent to the transcribed DNA. This too uses the DNA binding sites. Next one, the CRISPR-Cas9. The CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Unlike the ZFNs and TALENs, the CRISPR-Cas9 uses the RNA DNA binding sites. This makes it easier to engineer and can be used for a broad range of target sequences. They are derived from adaptive defensive systems of bacteria. Applications of genome editing. Better understanding. Scientists play with the genes in animals like mice and zebrafish to learn what all impacts a gene has on the animals and in turn us. This is because these animals have many of the genes as humans, and mice shares about 85% of the DNA with human. The next application is treating diseases. Genome editing can be used to prevent or treat diseases with a genomic base. There are two different types of genome editing, and they are germline therapy, in which the germ cells are edited and the changes passes down generation to generation. And the next one is somatic therapy, in which only the somatic cells are edited and only the individual is affected. Apart from these, our scientists face different problems like ethical concerns and technical barriers, but let's hope they find a way through them. I would really appreciate if you subscribed and liked this video. That's not it. Leave a comment on what you think about this video, or maybe how scientists can overcome the issues. Who knows if your idea is a solution? Only one way to find out. Bye.